Okay, can you hear me? Can I just do a sound check? If people can hear me? How do I make it? Yeah, there we go. Thank you very much. Thank you for your patience. We've got a good afternoon today to talk about our Oyster Translocation research results. My name's Mike Steer. I'm the Executive Director for the South Australian Research and Development Institute from the Department of Primary Industries and Regions. I just want to acknowledge that we ensure that land, sea, water and sky are healthy for future generations and strive to better understand the Aboriginal people's spiritual relationship with country across the state. In the spirit of reconciliation, our commitment is to build progressive and trusting relationships to share knowledge and learn from each other. We recognise and own a difficult past, and together we walk forward. We acknowledge the many Aboriginal people of this country as the oldest continuous living culture in the world, which is a great acknowledgement. First, we also acknowledge that it supports the White Ribbon Initiative and ending the violence against women and children. Thank you for tuning in today. Hopefully you can hear and see us clearly. The purpose of today's webinar is to provide you with an update on the results of the Pacific Oyster Translocation trials that we conducted to validate potential risks associated with transferring vital Karenia cells, or the algal species, and brevetoxin during the potential relay if oysters are from closed harvesting areas to open areas. So some really consequential results that have been research here. As you'll all recall, this has taken has been a very contentious issue with a number of risk assessments undertaken, one commissioned through the SAOGA, as well as by Person Biosecurity to help inform decision making at the time. However, industry were very keen for an experimental trial to be undertaken to validate these risk assessments. Now, these trials were funded under the Industry R&D program of the Algal Bloom Summer Plan, managed by the Department of State Development, and were conducted by SARDI from December 2025 to February 2026. A commitment was made to bring industry together to present the results and also to provide the opportunity for industry to ask questions of SARDI and the South Australian Seafood Quality Assurance Program, SASQAP, about the results and how they will be utilised in decision making moving forward. Today I'll have with me Dr. Sashi Nair, who's the Agriculture Program Leader at SARDI, who's led this research, and many of you would know, and will also provide an overview of the outcomes. I also have Sarah Sperling, the Acting General Manager of the Food Safety Biosecurity USA, and Emily Mellor, the General Manager Agriculture. And also with me, Emily's with me, and also have representatives from the industry as well. And there'll be an opportunity to ask questions of Sashi, Sarah, and Emily, and myself later in the program, if you go that way. I ask that you hold your questions to the end, please. There is a Q&A function on the webinar where you can post your questions as you think of them, and we'll try and work through them as many as we can as part of the panel discussion. Now, I'm going to give an overall update, maybe a little, some background, because I'm unsure what level of understanding the audience has. So I'll have a crack at getting everybody up to speed. Many of you would already understand this, and then we'll hand over to Sashi. So here we go. The South Australia's harmful algal bloom. So what we do know, and we have actually increased our level of understanding over the past 16 months considerably, and a real testament to the team here in South Australia and also the national collaborators. But the harmful algal species is a mix of Karenia species, and there's about half a dozen of them in the bloom. Historically, it was dominated by Mickey Motoy, and then we saw a change. I'll talk about that in a minute. But what separates Karenia species is known as a mixotrophic dinoflagellum. That means it can get its energy from photosynthesis, and also it means it can get its energy through organic materials that can consume on particulate matter. It has a couple of flagella or little tails, which means that it can move, and it can move quite quickly. which means they can go up to the surface during the day to photosynthesize and down at the bottom during the night to feed on other material. Some species, not all, in this complex produce biotoxins, and many of us are familiar with the brevitoxins which are underpinning today's conversation. They can impact fish, and the reason why they're called harmful algal bloom species is because not all of them are toxic, but they can still create levels of harm for gill-breathing marine organisms. So Karenia species can effectively clog up the gills. They can have various biochemical interactions with the gills. And also, if a bloom suddenly decomposes, it can draw a lot of the oxygen out of the water. And as a consequence, will have considerable fish mortalities or any marine mortalities that rely on gills to breathe. And indeed, we've got plenty of images that have reflected that over the year. There's also some public health concerns, particularly through mainly consumption of bivalves that accumulate the various biotoxins that Karenia species can produce, and that's why we've got the SASPLAT program in place. And there are other health concerns also for people who particularly suffer from asthma or have lung complaints or breathing complaints. If the Karenia species are aerosolized through storm wind action, particularly, and also if there's any brevitoxins in the air as well. So yeah, and as we all know, it's had a significant impact in South Australia, impacting around about 30% of the state's coastline. So this gives us a bit of a graphic of what actually happened. All right, so this is a image of the chlorophyll anomaly. So when you see these clouds of chlorophyll go up to red, it means it's high density. And the purple spots represent the cells of Karenia that have been validated. And as you can see from March, it's pushed off, ignited from Fleurieu Peninsula, pushed off through Baxter's Passage and Investigator Strait, working its way up through the Oyster regions of York Peninsula. It also sparked off over in Boston Bay and similar a little bit later and worked its way up the west coast. Spencer Gulf wasn't as impacted, but nonetheless still impacted as much as Gulf St Vincent. You can see here in Gulf St Vincent where it was hanging around the metropolitan coast, continuing through September, October. At this stage, we're getting large foaming events, huge amount of fish washing up. Now you can see it tracking around just before Christmas, the northern part of Kangaroo Island, down to the southern end of York Peninsula. The big swathes that you see down here is not bloom related. That's the upwelling that we would expect. It's important to understand that the chlorophyll in our water doesn't necessarily mean that it's harmful. In fact, a lot of the chlorophyll in there is a really good part of our ecosystem in terms of supporting primary productivity. But a good exercise, or at least illustration, to see the extent and persistence of what we now know is an unprecedented harmful algal bloom in Australia and probably ranking up in the top 5 in the world. Conditions have improved, but monitoring remains important. It's vastly disappeared throughout the majority of the state. There's a little bit of activity bubbling along Eyre Peninsula, and Eyre Peninsula are familiar with Karenia blooms, or at least the presence of Karenia micimotoi, and they have seen it for many years. And there is a little bit of activity around Port Turton, which is also dominated by micimotoi at this stage. We're expecting data to be uploaded later this evening. So if you're interested, jump on the dashboard towards 6 or 7 o'clock tonight for the update. So the questions that I quite often get is, what is it? What caused it? Can we fix it? And can we forecast it? So what is it? Largely a complexity of different Karenia species, and there's been some work done on the molecular diagnostics. Karenia cristata, the one to the left there here, is the one that produces the brevitoxins, and there are other various morphometric or different species on the right there. So what happened? This timeline's really interesting. So right at the beginning of the bloom, it was identified that the water samples that we took immediately were distributed to harmful algal bloom experts. They looked down a microscope and said, "Oh, it's Mickey Motorweg." Okay, so we thought, well, that makes sense. But then we started detecting brevetoxins in our oyster harvesting areas. Well, hang on a minute, but Mickey Motorweg doesn't produce brevetoxins, so it's clearly going to be something else in the mix. Then some work done at the University of Technology Sydney through molecular analysis identified cristata. So cristata is the guy that's producing the brevetoxin. We don't know a lot about this particular species. Then we went back and had a look at some of our historic information through our biomolecular observing, and we found that cristata as being a resident in South Australia as a natural part of our ecosystem since we started recording data back in 2016. So it's not a species that's been introduced. It hasn't come in through ballast water. It has been here the whole time at relatively low levels. When the conditions were aligned, it proliferated. So what caused it? Well, the information now, there's a report that's just been released today, actually, fortuitously. So you'll probably see some of this on the news this evening. Now, what caused it? We did say early on that there was 3 extraordinary environmental events that led to this bloom. The Murray River flood in 23, strong upwelling through the 23-24 summer that persisted through time. And then we had a heat wave, a marine heat wave in 2025. What caused it? Well, there was a whole heap of detailed work that's retrospectively gone back to find all the lines of evidence that we can get, or the data sources rather, covering satellite imagery, climate, nutrient data, upwelling indexes, etc. And then pulled it together and effectively said, well, these 3 massive events, the Murray River flood and the record upwelling, and those 2 rather, really primed the system in terms of productivity. We saw elevated levels of chlorophyll, we saw elevated levels of productivity, and the marine heat wave and the low level upwelling event in 2025 created warm, calm, layered, stratified conditions that were nutrient rich, which was the perfect Goldilocks environment for Corenia to proliferate, because Corenia loves those conditions and can outcompete other species that don't. Can we fix it? That's another one that comes up a lot. Well, there's a range of options out there in terms of physical, chemical, biological, and ecosystem-based methodology. But in general, these things generally apply to small, localised areas, and the science is not mature in many of those options. However, there is work that is happening concurrently through the Office for Algal Bloom Research that's looking into assessing the feasibility of these. But we're in general, worldwide, they tend to apply around confined areas, whether it's aquaculture production areas, whether it's ponds, dams, harbours, estuaries, etc., rather than applying it to the vast ocean and the various tidal and depth conditions that are experienced at an oceanographic or even at a Gulf level. Can we forecast it? Well, we're going to have a red hot crack at trying to forecast this. There's been a significant amount of investment into undertaking monitoring. We've got 6 of these big harmful algal bloom monitoring buoys that are distributed throughout the state. There's one at Copper Bay, Boston Bay, Stansbury, St Kilda, American River, Victor Harbour. There's a couple more smaller versions that will be implemented at some stage. But these buoys are picking up all the pedigree information that we're going to need for oceanographic modelling, plus they have real-time sensors for Corenia species and another species called Shatnella, which has also been a problem for finfish aquaculture around the world, and particularly around southern Eyre Peninsula. This is there's an example of the level of forecasting that we can have at this stage. This is relatively in its early stages of development, because this relies on particle tracking with a really greater depth of an understanding of the role that, or the biology and ecology of the different Corenia species, which is what we're working towards. Okay, this is a scary looking schematic, but this is essentially what we're aiming for, is pulling together all the information that we have through satellite imagery, all the information that we're gaining through our in situ monitoring, whether it's through our buoys or whether it's through active sampling or our research vessels or working with industry, pulling all our information around the biology and identification of the various species, integrating it with our understanding of the meteorological data, our oceanographic data, any of the environmental drivers. Some of that is maturing at the moment. And then pushing it through a series of multivariate sort of statistics and models to see if we can then effectively come up with a early warning system. So we can then be in a position to say, well, we think that a HAB has got a probability of occurring in this particular area at this particular time, which we can then inform industry quite early on to say, all right, well, now's the time to harvest, now's the time to change your operation. Community, you would expect to see potentially dead fish wash up. And who knows, maybe we'll be in a position later on down the track where we have a tool where we can go and mitigate or guard against it straight away. So this is effectively where we're working towards at the moment. There's some funding to support that. I'll whip through that really quickly. There's a lot of information, and there's a heap more other stuff that we're doing through the Office for Algal Bloom Research. It's established here, and the key to that, and as part of our role here at SARDI anyway, is to undertake research and development that will not only meet the needs of industry but also help develop the sustainable and economic production of our primary industries, and also help inform regulation, which is probably a really good segue into the next presentation. All right, I'll stop sharing and then I will pass over to Sheep. Thank you. Just bear with us as we. Oh, very well done. Thanks.
Thank you, Mike. Good afternoon, all. What I'll be presenting today is a large body of work that we undertook late last year, moving into this year, about the potential transfer of Karenia species and brevetoxins during oyster release from HAB-impacted farms. So this was a DST-funded oyster translocation trials with stakeholders from which included DST, Department of State Development, SEOGA, the Research Council, SASQAP, PERSA, Fisheries and Aquaculture. So the primary objectives of these trials were to understand the potential transfer of vegetative cells of Karenia species through translocated oysters from a HAB-impacted area. In this case, we got oysters from Kangaroo Island to a non-HAB-impacted area, which is Smoky Bay, when they were co-cultivated with each other in clean seawater. So that was one objective. The next objective was to evaluate the efficacy of the translocation treatment protocol on viability of Karenia, vegetative cells of Karenia species. And then the potential for active or passive transfer of bioaccumulated brevetoxins, which are the biotoxins from Karenia cristata mainly, from oysters originating from a HAB-impacted farm, in this case Kangaroo Island, into a biotoxin-free, non-HAB-impacted oysters sourced from Smoky Bay, co-cultivated in clean seawater at West Beach. Then to look at the residence time of bioaccumulated brevetoxins in live oysters from HAB-impacted farm, maintained in clean seawater, filtered seawater. There's not a lot of literature that exists on the residence time, so this was probably one of the most comprehensive trials that has ever been undertaken that answers many of these questions we have had for a long time. So the first question was, is the translocation management protocol effective in mitigating the risk of transfer of viable cells of Karenia from HAB-impacted oysters during oyster release? So the question that comes up is, what is the translocation management protocol for oyster relay? What is the prescribed protocol? It basically outlines that the oysters for relay needs to be washed, bathed, or hosed in potable water or filtered seawater that's been filtered down to 1 micron particle size. That is followed by a 24-hour drying phase in dark to desiccate the external surfaces at a temperature of less than 10 degrees centigrade and to clean a cool room. Then using clean baskets during the transfer, and then any equipment that is used for this purpose is washed down, which includes graders, baskets, bins, etc. So those are the 4 dot points which kind of outline the management protocol. So what we did was we basically looked at the oyster liquor as the first one to determine whether there were any cells present in HAB impacted oysters. And if you look at this table, we'll talk about uncleaned oysters versus cleaned oysters. Uncleaned oysters were not subject to the management treatment protocol. They were harvested and straight away they went into the cool room. The clean oyster, on the other hand, was subjected to the management protocol and then went to the cool room before being brought to Southwest Beach. So we looked at the oyster liquor as a first example, and then what we found was that the uncleaned oyster liquor sourced from Kangaroo Island had close to 3,700 cells per liter, plus or minus 1,604 cells per liter, with 3 replicates, which 16 oysters in each replicate. So we found that there were about 3,700 cells. When you look at the ambient SASQUAB data on the day, the seawater had 6,436 cells per liter. The same one, when it was subjected to the management protocol, we detected nothing in the oyster liquor, but we did see a large number of eggs. The logical explanation for this is that when you bring in unclean oysters, the moment you take them out of the water, the shells close, the 2 shells close tight, and they retain that liquor or the seawater that is trapped, and then they're only release it when they're exposed to seawater again. So in this case, the unclean oysters did not have that opportunity to release them, whilst the cleaned oysters was subjected to an exposure to water, and they either briefly, depending on if it's freshwater or seawater, and the duration of exposure, they either open briefly or and then they shut the shells. So that explains why we were seeing the cells in the oyster liquor. As one would expect, the oysters that originated from the non-HAB impacted area, which is Smoky Bay, we did not detect any cells either in the seawater or in the unclean oyster liquor or in the clean oyster liquor. Then we looked at what was growing epiphytically on the oyster shell surface. So for this, we scraped the surface of the shell and resuspended the material that we collected into clean filtered seawater and then counted them. So in this case, both the HAB impacted oysters or the non-HAB impacted oysters, both clean, whether they were subjected to the management protocol or not, had no cells whatsoever. We did, however, find some Dinophysis acuminata, one of the other HAB species, on the shells, but their numbers were very low. There were like 2,500 to 3,000 cells scraped from 36 oyster surfaces, which was not a big concern. General assemblage I've outlined there showed there were diatom, cyanobacteria, noplia, so there was an assemblage of other species on the shell surface, as one would expect. So the key take-home message from that small trial was that the risk associated with transfer of vegetative cells of Corenia from translocation through oyster liquor or as epiphytes on live oyster shells is quite low. We do recommend additional farm trials to remove confounding factors that influence this trial, and I'll talk about that in a minute, and also to determine the blooming potential, which we couldn't do when we did this trial at short notice. So the recommended improvements to the study includes repeating the control study to test the efficacy of the management strategies. And the key weaknesses of the current trial included the transportation time was not standardized. We brought back oysters from Smoky Bay that took, and also oysters that we moved from Kangaroo Island to West Beach, and these times were not standardized. And as a result, that was identified as one of the confounding factors. This was done at the peak of summer, around the 9th of December. So we did expect that the oysters would be stressed during transport under high ambient temperatures. Spawning risks were another one which we did encounter during this trial. And the suggested improvements to this includes was identified to be doing this trial on a farm when the Karenia counts were high. So rather than bringing it to West Beach to do it straight on the farm to remove any confounding factors with regards to transportation time and handling. And also the Karenia being very delicate, it's recommended that we undertake the counts soon after collection on farm without undertaking any movement whatsoever. The trial to be extended to ascertain the blooming potential, which means whatever potential vegetative cells might be growing either epiphytically on the shells or residing in the oyster liquor, by cultivating them in a growth media for about a week and seeing whether they tend to bloom or not. And then, of course, increasing the replication. These trials have, or what has been proposed, the improvements that have been proposed have been funded by DSD. Should the Karenia counts ever increase to more than 5,000 cells per liter in any of the farms, oyster farms, over the coming years. The next question was, is depuration a pathway for transfer of viable cells of Karenia from HAB-impacted oysters during a filling? The depuration pathway is mainly what goes through the gut passage and comes out, and that was one of the other considerations as part of this trial. So what we did for this trial was we co-cultivated clean HAB oysters or oysters sourced from a HAB-impacted area, that is Kangaroo Island, with non-HAB impacted oysters from Smoky Bay in triplicate for 1 week. We then ascertained the Karenia in seawater, where this trial was undertaken, both in the morning and in the afternoon, which is twice daily. And these were the setups. So we ran them in triplicates. We had 3 large tanks with a small impeller to keep cells in suspension, and also to recirculate water, to circulate water within the tanks. And this was the setup that was used, and this was sampled morning and afternoon for Karenia counts. So that's another photograph of the experimental setup. From day 0 to day 9, no Karenia cells were detected either in the morning samples or in the afternoon samples in either of the feed replicates. We did, however, find that the assemblage was dominated. There was an assemblage of other planktonic species. It was dominated by Copypod nauplii, marine worms, diatom, cyanobacterial filaments, egg masses, oyster eggs, flagellates, seaweeds. So it was not devoid of life. It very much was, but no Karenia was detected, which is a promising sign. So this is how the Karenia counts were undertaking. Large number of samples came back to the lab. We then followed the SASQA protocol of concentrating the sample and then counting them with the centrifugal after cell, and triplicate counting of each sample was undertaken to remove any potential errors in counting. So the key take-home message from this trial was that the risk associated with transfer of vegetative cells of Karenia through depuration, that is through the gut passage, of HAB impacted oysters subjected to translocation management protocol is low. Then we looked at the brevetoxin aspects of the algae. So the short-term and long-term dynamics in brevetoxins and its analogs in oyster tissue and oyster liquor. Now, just to give you a bit of a background on brevetoxins. Brevetoxins cause neurotoxic shellfish poisoning, which is a foodborne toxicity from the consumption that comes up from the consumption of brevetoxins or brevetoxin analogs, largely through seafood. These class of compounds or the brevetoxins are thermally stable and they're lipophilic, which means they show a tendency to bind to fats or lipids, and they are not inactivated by cooking or freezing, which makes it extremely challenging to counter that. Now, brevetoxin 1 and brevetoxin 2 are the two parent toxins that are biosynthesized by the marine dinoflagellate. Now, what is important to note, and I put an asterisk there, is brevetoxin 1 is not synthesized by the species of Carenia that we saw here in the South Australian algal bloom, which is Carenia cristata. Carenia cristata does not produce brevetoxin 1, it only produces brevetoxin 2. On the other hand, the one that you see in the Northern Hemisphere, especially in North America, which is Carenia brevis, produces both brevetoxin 1 and brevetoxin 2. So shellfish, particularly oysters, tend to bioaccumulate brevetoxin in tissue through filter feeding. So they filter feed Carenia cells when they're in large numbers, and then tend to bioaccumulate the brevetoxins in tissue. In the shorter term, this does not cause any major issues to the oyster. They just accumulate. But in the longer term, we have seen some impacts to oyster. We're talking about beyond 9-10 weeks. And neurotoxic shellfish poisoning is characterized by gastrointestinal and neurological symptoms that include vomiting, diarrhea, numbness, tingling in facial regions and extremities. So no fatalities have been recorded from NSP yet. However, there was a mass toxicity event of over 180 people that occurred in 1992 in New Zealand, associated with the consumption of brevetoxin-contaminated cockles and green mussels. And the toxicity effect is through brevetoxin binding to the voltage-gated sodium channels on cell membranes, opening the cell pores, resulting in an influx of sodium ions into the cell and causing the excitation of the nerve impulses, which is the way it impacts on in humans and other animals. So the Food Standards Australia and New Zealand, FSANZ, has recommended the regulatory limits to be around 200 mouse units per 100 grams of shellfish tissue. That translates to approximately 0.8 milligrams of brevetoxin 2 equivalent per kg of oyster tissue. And this is how it's measured. The output that we get in terms of concentration is what we get out of an LC-MS, which is the instrument that we use for measuring brevetoxins. So just to give you an idea as to what the metabolic transformation processes are of brevetoxin 2 and the production of the various analogs, I'll just go through this little schematic. So as I mentioned earlier, Carenia brevis produces brevetoxin 1 and brevetoxin 2. They are biosynthesized by the algae, microalgae. We do not see brevetoxin 1 in Carenia cristata. So Carenia cristata was the main HAB former in South Australia, and we do not see brevetoxin 1 production. So brevetoxin 2, and what I've so I've put 2 stars. The red star indicates that it's been reported in literature that is common in Pacific oysters, and the purple star is based on the persistence in this trial and also in the field observation. So brevetoxin 2 is commonly found in Pacific oysters, which is biosynthesized by the algae. When it undergoes oxidation, it produces the analog brevetoxin B5. which is also common in Pacific oysters. When this combines or gets conjugated with taurine, which is an amino acid, it produces brevetoxin B1. Noting that all analogs of brevetoxin are basically metabolic analogs during the various transformative processes, where there is oxidation, reduction, conjugation, or cometabolism in oysters. When brevetoxin 2 undergoes reduction, which is an aldehyde group in brevetoxin 2 gets transformed to an alcohol group, it produces brevetoxin 3, which is a persistent brevetoxin that we find. It's waterborne, and we've been seeing this not only in oyster tissue, but also in oyster liquor, and also in field-based observations, farm-based observations. When brevetoxin 2 undergoes cysteine, another amino acid, it performs brevetoxin B2, which is one of the other analogs. Brevetoxin B2 is also a persistent analog and is also commonly noted in Pacific oysters. When this further goes through conjugation with fatty acids like myristoyl and palmitol, it forms brevetoxin B4. We do find at times, but it has not been a persistent form. Brevetoxin B2 further goes through a metabolic transformative process that involves microbes within shellfish, and that's a process called as cometabolism, and that produces a very common brevetoxin analog, which is S-deoxy brevetoxin B2, which was not only known to occur in Pacific oysters, but was also observed in this study as being persistent, and was one of the most dominant analog. So then, with that background in mind, the question was, is there a difference in brevetoxin concentrations and its analogs in cleaned and uncleaned oysters? When I say cleaned, again, cleaned was subjected to management protocol, whereas uncleaned was not. So we looked at the total brevetoxin concentrations here, and in both oyster tissue and oyster liquor. And note the benchmark, which is the dotted red line, which is a 0.8 milligrams per kg of total brevetoxins as recommended by FASANS as the safe regulatory limit. The green box should have been Smoky Bay. Obviously, there was nothing detected in Smoky Bay. That's why nothing is showing up in the graph. And Kangaroo Island is denoted in orange. So when we looked at cleaned versus uncleaned in oyster tissue, we did not see any statistically significant differences for 3 replicates. And that was at a level of between 2.14 milligrams per kg was what was recorded, which was about which was significantly higher than the threshold, recommended safety threshold. On the other hand, when you look at the oyster liquor, there was hardly any. It was very close to limits of detection. So it could be safely said that oyster liquor hardly had any brevetoxin in them. When you look at the analogs, the oyster tissue, the analogs in the oyster tissue were very comparable between clean and uncleaned oysters. So, and largely dominated by S-deoxy brevetoxin B2, brevetoxin B3, and brevetoxin 2, which is what I mentioned earlier. There's maybe subtle differences in some of the levels of the analogs, but overall, that was very comparable between the two. Oyster liquor, on the other hand, hardly, there was absolutely very low levels of various analogs detected, and it was not worth teasing out any further. In summary, no brevetoxin was detected in oyster tissue or liquor of cleaned or uncleaned oysters from Smoky Bay, which was to be expected because that is supposed to be our clean site. No significant differences in tissue brevetoxin concentrations of cleaned and uncleaned oysters from Kangaroo Island was noted. So the mean brevetoxin concentrations varied between 2.02 milligrams per kilo to about 2.14, significantly higher than the prescribed safe limits of 0.8 milligrams per kilo of oyster tissue. Tissue brevetoxin concentrations were about 2.5 to 2.7 times higher than the safe threshold of 0.8 milligrams per kilo. In liquor, it was about 35 to 40 times lower than the tissue levels and was always below the regulatory limits. The brevetoxin analogs in cleaned and uncleaned oysters were quite comparable, except for the presence of brevetoxin B1 in cleaned oyster tissue. The dominant brevetoxin analogs were S-deoxybrevetoxin B2 and brevetoxin 3 in both cleaned and uncleaned oyster tissue. So when you look at the analogs, they were very comparable in oyster liquor, but again, they were very, very low in terms of magnitude, almost close to detection limits of the instrument. So the next question was, do brevetoxin transfer from cleaned Hab impacted oysters to clean non-Hab impacted oysters when they are co-cultivated for 8 days, which is a very short-term study to look at in the short term, do they transfer between Hab impacted and non-Hab impacted oysters. So these were the oysters that we got from Kangaroo Island and Smoky Bay. They were transferred into oyster baskets, clean oyster baskets, and they were then maintained in the large tanks that I showed earlier. On the right is the panel that you see for oyster tissue. On the left, on the right is what you see for oyster liquor, and on the left is what you see for oyster tissue. And as I mentioned earlier, the Smoky Bay is in green and Kangaroo Island, which is the Hab impacted site, in orange. Now, we did notice a very small amount of brevetoxin that transferred, but it was minuscule to the point that it was very close to detection limits. So as you can see, within a span of 1 week, there was a significant reduction from about 2.14 milligrams per kilo to around about 1.02 or so milligrams per kilo, 1.2 milligrams per kilo, or within a week. So that's about a close to a 50% reduction in brevetoxins in oyster tissue. In oyster liquor, again, the same story. There was hardly anything detected, very close to detection limits of the instrument. When we look at the analogs, it was the same story again. There was some small changes in brevetoxin analogs, but the dominant ones being S-deoxybrevetoxin B2, brevetoxin 2, and brevetoxin 3 being the dominant ones. Again, in the oyster liquor, there was hardly anything detected. There was, there was some detection, but it was close to limits of detection instrument. So the key summary from this short 8-day co-cultivation trial was that we did notice a 50% reduction in brevetoxin concentrations in tissue over 8 days in Hab impacted oysters. This reduction was about 20% oyster liquor, but oyster liquor to begin with was very small anyway. The tissue transfer of brevetoxin from Hab impacted oysters to non-Hab impacted oysters was insignificant after 8 days of co-cultivation. And even if you looked at the profile, it was mainly brevetoxin 3 that was transferred. But again, it was not significant. The total brevetoxin concentration in liquor was about 35 to 110 times lower than tissue, which explains that pathway for any movement of brevetoxin can be safely ruled out. Small concentrations of brevetoxin that transferred from Hab to non-Hab impacted oyster liquor at the end of was very small at the end of 8 days. Mostly brevetoxin 3 with smaller concentrations of S-deoxybrevetoxin B2 and brevetoxin B2. So the brevetoxin analogs varied in tissue at the end of 8 days. There was a decline in brevetoxin B5 and S-deoxybrevetoxin B2. But again, we saw a 50% decline in total. Oh, slides are frozen. Presentation slows.
And I think it's moving on here.
Moving there, it's not moving on the screen.
There you go.
Stop.
Yeah.
No, it's not moving.
So
It's,
Shut it down, start again, mate.
okay,
I think it's.
okay, okay.
Just got to catch up. We've had.
Okay. So the key questions were, the next set of questions were, do brevetoxin transfer from clean HAB impacted oysters to clean non-HAB impacted oysters when co-cultivated for 9 weeks? That was the longest duration. That's a long duration study that we did. And then that study also answer was, we had the question on what is the residence time of brevetoxin in HAB impacted oysters that declined and how long it took to decline to regulatory limits of 0.8 milligrams per kg when co-cultivated, when cultivated in HAB-free conditions. So that is like simulating a potential limit. So what we did was we co-cultivated clean HAB oysters, that is one subjected to the management protocol, sourced from Kangaroo Island, with non-HAB impacted oysters sourced from Smoky Bay in triplicates in tanks for 9 weeks, that's 63 days. We monitored Carenia in seawater. In fact, the samples were collected daily, but brevetoxin samples were only collected weekly. And the weekly variation in brevetoxin concentrations in HAB impacted tissue and liquor was noted and was measured. So this was one of the 3 tanks that we used. And you can see these were HAB impacted oysters. Equal number of HAB impacted oysters were put, cleaned oysters were put in baskets, and they were co-cultivated with non-HAB impacted oysters sourced from Smoky Bay in the same tank. The way we ran this trial was with feeding. So early in the morning, every day, early in the morning, we would take water quality measurements, collect a Carenia sample, and then we would drain the tank to simulate what happens in a farm to replicate a low tide. And then that stayed for 4 hours in a subjected to air drying. The oysters were air dried. After 4 hours, we started gradually filling up the tanks. And during the process of filling up the tanks, which simulated a high tide, we would add feed, which was in the form of either paste, algal paste, or live algal cultures. Bottled pastes were used for this on alternate days. So this was the water photograph showing the water quality sampling in the morning, Carenia sampling in the morning, and then on alternate days we were feeding with live algae and bottled algal paste. However, we were noticing that the live algae was a lot better preferred. So once we got to a stage where our live algal cultures in a hatchery grew to a substantially large volume, we pretty much started using live algae over bottled algae to make sure that it was that we could run the trials in the best conditions. So this is what the mixed live algae production was in the hatchery under controlled conditions, and then we would drain it out into this big tank, and then we would feed them to the oysters in the pool farm. And every week, we would then take the sample, subsample the oysters, and we would shuck it, and then process the oyster tissue by homogenizing it. And the homogenized samples were then shipped to Cawthron Institute in New Zealand. And later in the year, we started shipping it to Agilex. And we did the same thing after shucking. Whatever oyster liquor was collected, that was also sent for analysis. So the results were that this is the 9-week result. As you can see, this is for the tissue. And obviously, Smoky Bay is plotted with Kangaroo Island. There was nothing in Smoky Bay. So therefore, nothing was transferred to the Smoky Bay oysters all through the 9 weeks. Kangaroo Island oysters, the HAB impacted oysters, started at about 2.14 milligrams per kilo, and that was on day 0. And then that reduced to below the regulatory limits on the first week of the trial. However, on week 2, it did go up marginally, and that's a similar trend that we see in wild oysters too. And then subsequently, it declined after week 3 onwards to below the regulatory limits of 0.8 milligrams per kilo. The only logical explanation for the week 2 spike could be that it was probably in a analog that's not currently measured by the LCMS technique, that may have transformed into one of the other analogs, most likely S-deoxybrevetoxin B2 after week 2 onwards. As you can see, the dominant brevetoxin analogs were S-deoxybrevetoxin B2, which is the most dominant, followed by brevetoxin B2, and a small amount of brevetoxin 3, which is the transformative product, which is the one that's water reduced. So that's the plot showing the changes in brevetoxin analogs over time across 9 weeks. As you can see, there was nothing detected in Smoky Bay oysters. It was all in Kangaroo Island oysters, and that declined after week 3 onwards. So as you can see, the dominant one analog that was present all through the trial was S-deoxybrevetoxin B2, followed by brevetoxin B2. So in summary, we're seeing a 75% reduction in total brevetoxin concentrations in tissue in the first week, followed by a small spike in week 2, followed by a decline to levels below threshold from week 3 onwards. There was no tissue transfer of brevetoxin from HAB impacted to non-HAB impacted oysters all through the trial, from day 0 to week 9. There were only minor changes in brevetoxin analogs that we observed over tissue in tissues over time. After day 0, the changes in brevetoxin analogs were very subtle. The dominant ones being S-deoxybrevetoxin B2, which is the most dominant, accounting for about 40 to 60% of the total brevetoxins, followed by brevetoxin 3, which is about 10 to 30%, and brevetoxin B2, which is about 10 to 20%, and a very small fraction of about 5% of brevetoxin 2, which is the biosynthesized fraction in oyster. When you look at the liquor, there was hardly anything. So it was as good as close to being close to the detection limits. After day 0, there's very little detected or which was insignificant. So that kind of is an interesting story. And when you tease it out a bit more, again, it is immaterial because in oyster liquor, the overall concentrations were very low, but it's largely made up by S-deoxybrevetoxin B2, which then transformed to brevetoxin B2 by week 4, after which nothing was detected. So in summary, the brevetoxin concentrations in the liquor was about 40 times lower than in tissue. Again, we can safely rule out that this was one mechanism for transfer, which we initially thought before. At the time of the planning this trial, we thought maybe liquor was the major vehicle for transmission of brevetoxins. But that's now been conclusively proven that it's an insignificant factor for transfer. A 10-fold reduction in brevetoxin concentrations in oyster liquor was observed in the first week. There was no brevetoxin transfer through liquor of HAB impacted to non-HAB impacted oysters. And the significant changes in liquor over time, there were significant changes, but again, the levels are 15 times below the safe threshold limit. So it's not a key consideration. When you look at the tissue, the total brevetoxin concentrations in the tissue fell below the safe threshold levels from week 3 onwards. That is 21 days. There was a minor spike in week 2, after which it declined and stayed below the threshold, following the trial gradually declining over the period of 9 weeks. There was no tissue transfer of brevetoxin from HAB impacted to non-HAB impacted oysters at any time scales. Either the short 8-day trial or the 9-week long trial, we did not observe any tissue transfer from HAB impacted to non-HAB impacted when they are co-cultivated together. And that is of relevance from a relay perspective. Changes in brevetoxin analogs in tissue over time is minor. The profiles were relatively stable, and that is very much in line with what has been reported in the literature, the few studies that have been done on Pacific oysters. As I mentioned before, the oyster liquor is not a major vehicle for brevetoxin mobilization in oysters. So the key aspects to consider from this study is that this study was undertaken when the HAB impacted oyster tissue registered a total brevetoxin concentration of 2.14 milligrams per kilo, which was the level when it was sampled from Kangaroo Island. The decline in tissue brevetoxin over time and potential transfer from HAB impacted to non-HAB impacted oysters are all based at the threshold level of 2.14, because that's the only oysters we could source at the time of the trial. The key question is whether this trend is comparable if tissue concentrations of HAB impacted oysters reach between 20 to 30 milligrams per kilo, as observed in certain farms during the peak of the HAB. Stansbury is a good example of that, where it had reached around close to 20 milligrams per kilo at the peak of the HAB. Could be extrapolated. What could be extrapolated? This trend could be extrapolated from some of the depuration trials that have been undertaken. But then we got to keep in mind that depuration trials involve no feeding whatsoever, and is not comparable to a relay scenario into a classified area, because they're getting fed natural food that comes with the tides. So the depuration trials obviously do have an impact on oyster condition if it is done or undertaken over a long period of time. So the key question there was, what happens if the total brevetoxin concentrations go beyond 2.14 milligrams per kilo? So obviously we didn't have that any samples from that we could use in our experimental trials. So then we looked at the extensive data that SASQUAP collected from various classified areas during the peak of the South Australian bloom. So these are the observations from the 5 farms. And the key objectives of this trial or this study was to assess the rate of reduction of brevetoxin from HAB impacted farms with the peak brevetoxin concentrations of up to 20 milligrams per kilo. Then we wanted to look at the relationship between Carenia cell counts in real time and how that translated with brevetoxin concentrations in tissue from various from the 5 farms that we chose as the sample farms or the classified areas. Comparing the brevetoxin analogs or the profiles from HAB impacted farms corresponding with brevetoxin peaks. So the first one that we looked at was Kangaroo Island, logical because American River, which is Todd Packer's farm, was one that we used. Oysters from there was used for this experimental trial. So it was logical to look at that. What we found was there was a very clear-cut delay between peak of Carenia blooms to the peak of brevetoxin concentration in the oyster tissue, and that is to be expected because they are filter feeders. So when there's a peak of Carenia in the water, they would be filter fed, and eventually they do bioaccumulate, and that's reflected with that short lag in concentrations. What we did observe was that it took about 6 weeks from the peak of the brevetoxin concentration in the tissue for that to get down to the regulatory levels of 0.8 milligrams per kilo. When you look at the profiles, the brevetoxin analogs, again, mirrors what we saw in the experimental trial with S-deoxybrevetoxin B2 being the most dominant, followed by brevetoxin B2 and also brevetoxin B4. I'll summarize that in a minute with all the other farms. Stands per day. This was of most interest because the concentrations there peaked up to 20 milligrams per kilo. And the cell concentrations reached peaks of about 800,000 cells of carina per liter. Again, a very similar observation where the carina peaks, where the brevetoxin peaks followed carina peaks. In this case, it took about 19 weeks for the tissue concentrations to drop to regulatory levels of 0.8 milligrams per kilo. That was a really good observation because we know that at 2.12 milligrams or 2.14 milligrams per kilo in the experimental setup, we were able to see that reduction within 3 weeks. But when it gets to about 20 milligrams per kilo, from the farm observation, we are seeing that it took about 19 weeks. Again, the analogs were very comparable. And then we looked at the classified area of Port Vincent. And in that particular farm, again, the similar observation, but the peaks of the brevetoxin peak was around 13 milligrams or 12 point something milligrams per kilo. Took about 14 weeks for that to reduce to the regulatory levels of 0.8 milligrams per kilo. Again, the analogs were very comparable, dominated by S-deoxybrevetoxin B2. Franklin Harbor, we selected 2 farms as SWB, 2 classified areas, SWB2 and SWB9. And that's where the location of the 2 classified areas are. In this case, when we looked at SWB2, the peak brevetoxin concentrations were around 1.9 milligrams per kilo, and that took about 4 weeks to decline to the regulatory levels. Profiles were comparable. When you looked at SWB9, which is an adjacent classified area, the peak brevetoxin concentrations reached 2.5 milligrams per kilo, and that took about 7 weeks. Noting that this is likely to be slightly different from a relay scenario or from the experimental setup, because the carina cell still present in the womb. Oh, sorry. So when you look at the brevetoxin analogs or the profiles during in various farms at the peak of brevetoxins, the profiles were very comparable, except for American River, where there was a slight difference seen with brevetoxin B1 showing up in the profiles. None of the other farms showed that. That could be explained due to 2 reasons. One is that is due to potentially 2 reasons. One is that was a time when we transitioned from Cawthron Institute to Agilex for the analysis. So there were subtle difference in the reporting limits, or it could also be due to the composition of the phytoplankton that they were filter feeding at Kangaroo Island, because this is largely due to the taurine. This is the taurine conjugate. Other than that, when you compare the profiles of the analogs, they were very comparable across the various farms, various classified areas. 2 in Gulf St. Vincent, 1 in American River, and 1 in and 2 in Spencer Gulf, Upper Spencer Gulf. Just clearly shows that regardless of where it's spatially located, the tendency or the reproducibility of the trends are very similar. So we put all that in a table summarizing, and that's when we observed some interesting trend with peak brevetoxin concentrations, with the duration it took for that peak to decline to regulatory limits of 0.8 milligrams per kilo. So this is the summary table, and what we thought we were interested in looking at was, can the duration of brevetoxin concentrations to decline to regulatory limits of point or less than 0.8 milligrams per kilo be predicted based on these observations? And the answer is yes, we could. And we saw a really good correlation when we plotted that observation. And with an R square of 0.901, which we were not expecting. This was an accidental observation. But what we got out of that was quite valuable. So when we use that predictive model to plug in various concentrations as observed from the various farms of the experimental results. Now, with the experimental result, the first one that you see on the top, 2.14 milligrams per kilo, was what was observed in Kangaroo Island, what was brought to West Beach. As per the predictive, when you plug it into the regression equation, the prediction says it would take about 3.8 weeks for it to decline to the regulatory levels of 0.8 milligrams per kilo. But experimentally, we validated that it took 3 weeks. So there's a really good correlation there. So it's very comparable, probably a bit more conservative. When it got to about 1.83, it took about 3.5. It estimates that it takes about 3.5 weeks. Once it increased to about 19.94 milligrams per kilo, as observed in Stansbury, it predicted that it will take about 18.9 or 19 weeks for it to reach the regulatory limits of 0.8 milligrams per kilo. So the key observations from this farm observations or the classified area observations were there's a lag between Corenia cell counts and brevetoxin concentrations. The changes in brevetoxin analogs in oyster tissue during brevetoxin peaks from various HAB-impacted farms is minor. Even though they are very spatially spread, that change in analogs was minor. Persistence of brevetoxin in all concentrations in oysters from on-farm observations in the presence of Corenia cells is a lot longer, as one would expect, than in experimental observations where we don't have any Corenia in the background or in a depuration trial. On-farm data clearly demonstrates a linear relationship between brevetoxin concentration and the time it takes to reach safe levels of 0.8 milligrams per kilo. This could be regarded as more conservative than in a relay situation where one would expect that there will be no background levels of Corenia at all or any brevetoxin presence in the water, sediment, or pore water. Experimental data is very comparable to oysters relate from a HAB-impacted farm or a HAB-impacted area to a non-HAB-impacted area, because there would be no background levels. So in conclusion, the oysters subjected to translocation management protocol did not register any Corenia cells, either growing epiphytically on the shell or in the oyster liquor. No Corenia cells established when uncleaned and cleaned oysters were co-cultivated for 1 week, that 8-week trial. Brevetoxin concentrations in oyster tissue during the experimental trial declined to regulatory levels with 3 weeks at initial levels of 2.14 milligrams per kilo. The concentrations in oyster liquor is negligible or close to detection limits in all the trials. So we can safely rule out oyster liquor to be contributing in any way, shape, or form in brevetoxin transformation, or brevetoxin transfer. So during 9 weeks of co-cultivation, no brevetoxin transferred from HAB-impacted oysters from Canberra Island, co-cultivated with HAB-free oysters from Smoky Bay in our trials. The changes in brevetoxin analogs in oyster tissue during the trial was negligible. On-farm observations clearly demonstrate a lag between peaks of Carenia cell counts and brevetoxin concentration. Brevetoxin analogs in oyster tissue corresponding with brevetoxin peaks from various HAB-impacted farms is comparable. The persistence of brevetoxin in oyster tissue from on-farm observations in the presence of Carenia cells is longer than experimental observation, which includes depuration and tissue washout. Depuration in the first few days, but tissue washout occurring subsequently. On-farm data demonstrates a linear relationship between brevetoxin concentrations and the time it takes to reach regulatory levels of 0.8 or decline to regulatory levels of less than 0.8 milligrams per kilo. Higher the brevetoxin concentration, the longer it takes for it to be washed out from tissue, as we have now seen with those observations. I would like to acknowledge the project steering committee from DST, SEOGA, the Research Council, PERSA SASQUAP, PERSA Fisheries and Aquaculture, and Richard from SADI. There's been a lot of input into this trial over the duration we used to meet when we met, and that has shaped this trial to where we have come. The experimental trial that was undertaken at SADI commenced on the 9th of December. We went through the Christmas break, over New Year, monitoring daily until it finished in mid-Feb. And I want to acknowledge my team from SADI. Funding support from Alder Bloom Industry Research and Development Program. Oysters for the trials, Todd and from KI Oysters, Joe and Linton from Smoky Bay Oysters. There was a lot of logistics involved in moving them across during the peak of Christmas. And they were absolutely helpful in ensuring that we got those oysters in time to run these trials. And we thank them greatly for that. Farm data, Ali from SASQUA provided that, and we put that in a presentable format, and that has formed a large basis of some of these conclusions. We did seek biotoxin advice from Tim from Cawthron Institute, who's also contributed substantially in giving advice on how the results were interpreted. And the brevetoxin samples were analyzed at Cawthron Institute and Agilex. We still have water samples that we'll be doing at SADI down there. That brings me to the end of the presentation. Thank you very much. And that's a beautiful photograph from Coffin Bay on a beautiful sunny day.
Thank you very much, Sasi. That is a comprehensive body of work there, and I acknowledge that there was quite a lot of information that was disseminated relatively quickly. So we will distill that information into a simple summary and also pull together the slide set to distribute in good time. So you can then revisit that. I think the recording will also be provided so you can get a little bit more of an interpretation from the slides, which can help you distill that information. And as always, you know, SADI industry are connected well and PERSA if we need to answer any more questions going forward. But now there's an opportunity for people online to ask questions now, and you know, we're here for another half an hour or so. They are coming through. I can read them out. You put them in through the Q&A, and we'll go through them accordingly. First one is, when assessing risk of translocation protocol efficacy, what was the cell results from a non-translocation protocol control? Asked another way, there were no Carenia in 0 to 9 days. What was the cell counts in the control outcomes?
The cell counts at the time when we sourced it from Kangaroo Island was close to 6,000 cells per liter. That was the background level. And then once it was brought in, then it was maintained in clean seawater.
Yeah, you have that one.
Yeah, right. Thank you. We'll be patient for anyone who would like to type in acknowledging that, yeah, that was a lot of information that we delivered today. Yeah, and that, we will monitor both the Q&A, but also the chat, which a lot of people are familiar with. No stress, that's fine. We will distribute the information.
And we're happy to chat with Lindley as well, whether there's an opportunity to provide a much more condensed version of that at the October Oyster seminar, if you feel that there'd be value in either hearing that again or potentially for others that haven't made it today to have the opportunity to hear it too.
All right, well,
It's very quiet
I'll
today.
give it 30 seconds. Oh, hang on. Up and away.
This one is
All right, okay. Is there an estimate on how long it will take transfer protocols to reflect this information?
It's from what we've seen with the information from this study, the information we request for a relay request would remain the same, and the relay process would remain the same.
That question. Any answer to that one? Okay. Another one here. Am I right in recalling the Corenia cell counts from KI were below translocation level anyway when conducting this? What would counts over 1 million cells contribute, i.e. as per full HAB highest levels? What were other diatom levels in the same time?
So we don't have a limit or a number of what level of Corenia would see a closure of an area. So we would only take into account the protoxin levels in closing area. The level of Corenia, we don't have any numbers that would impact how we would manage a relay. I think from what the research has started showing that once it's gone through that management protocol of the cleaning process and stored out of water for 24 hours and those 4 points that Sashi had mentioned, the risk is low.
There's another question there. What were other diatom levels in that at the same time?
We got the data. We haven't really looked at the diatom levels for this presentation, but that's something we can certainly will be looking at down the track. But we have the community structure of the pilot plant.
No more coming through, and that's okay. There'll be plenty of opportunity for us to reply to any questions that come through our session. So if that's the case,
There's just a couple of questions around the relay protocols. Guys, have we clarified that for you?
so that question that Sarah answered, can you confirm that the relay protocols will remain unchanged?
So the relay request form is on the website, and we'll send that link out with the key points from the summary that we put together with that. As part of the relay request, there is information that we'll require on where are you going to locate the stock, how are they going to be quarantined. There is a need to test stock that has been relayed to release it. Now that is the standard process. The implementation of that standard process prior to this has been improved. So that's probably the change that the industry will see in terms of whether it's for biotoxins or E. coli. If you're going through a relay process, the need to test relay stock. That's something that has come from the national review that DAF undertook of the relay process. So that's something that we will be overseeing.
So just so I'm hearing that right, there's an available online, the protocols that industry need to adhere to. So it would make sense to pull that out and package it in this so it's consistent with the topic of the day.
Absolutely.
Yeah.
And I guess the one thing that this research has given us more visibility of is potentially how long it might take for a relayed oyster to then return to those, so under that 0.8, to be able to, to be suitable for sale. So, as you've seen, there's that direct correlation. So those that were sort of, much higher in the brevet toxin content of the oyster will potentially take much longer to return to that 0.8 level and be able to be sold. But what it does do is give us that confidence that we could potentially move those oysters and they could be in an area, and at least they're able to be, kept in their environment. They're able to continue feeding, as long as they're able to be kept separate from the other oysters for testing purposes. Then you'd have to just wait for that period for the brevetoxin to clear, and at least you have them out of the environment that they were accumulating more brevetoxin.
Will the testing also be required for relaying of juvenile stock?
To move that stock out of its quarantine status as part of its relay, yes, but I will seek clarification from DAF on that one if we have any options around that, but I'd say yes.
Yes, but we'll see clarification.
Yeah.
I'm assuming that the idea behind that is that juvenile stock would not be
Not suitable for sale
Yeah.
for over 60 days. Yeah. Well, juvenile stocks.
When we are talking relays, you're referring only to a harvest area under closure or being currently affected by a HAB.
Only those areas that are closed. If an area is impacted by HAB but hasn't closed, stock is free to move.
Will other industry stakeholders within Bay areas be aware of any relocations? Or will it be available within industry via some way? How will that be communicated?
There's that one for you.
So if a relay is approved,
Yes, will industry stakeholders be aware that a relocation has been approved at relay transportation?
it's not our protocol to notify anyone other than the person who's relaying the stock and the accredited producer who's receiving the stock.
So that's the protocol. All right. I'll stretch it out for 30 seconds to see if there's any other takers, acknowledging that we can catch up again at a session. Going once. Yeah, it's hard, and it's awkward because we're looking at ourselves on a big screen. So, thank you for everybody who participated. Like I said, a lot of content that'll be summarised and distributed, both in summary form and in full form, plus the associated procedures that are relevant. Really appreciate your time. I feel like this is a good platform to disseminate key bits of information to ensure that the scientists, the regulators and industry are in the room. I think it's a good format. So thank you very much. We will draw this to a close and look forward to connecting with you again another time. Enjoy the weekend. Crow's already lost, so I've got nothing to do with it. Thank you, everyone.
Thanks, everyone.